Welcome back to the fifth video reviewing the basics about T cells and why we love them. Hi, I'm Christopher. So far, we have talked about how T cells see the world, how the thymus educates T cells, how MHC displays peptide evidence, and how CD4 and CD8 T cells perform different jobs. Now we're going to ask another important question. If the body has many T cells, how does the immune system find the one that recognizes a specific antigen? That is the idea behind clonal expansion. Before infection, the body already contains many different naive T cells. Each one has its own T cell receptor, or TCR. Most of those T cells will not recognize a particular pathogen. But somewhere in that huge crowd, there may be one rare T cell whose receptor matches the peptide MHC complex being displayed. Clonal expansion is a process where that rare matching T cell is selected, activated, and expanded into a larger army of antigen-specific T cells. In simple terms, one rare match becomes many responders. Let's start with the challenge. The immune system has an enormous diversity of naive T cells. That diversity is a strength because it means that the body is prepared for many possible antigens. But for any single peptide MHC target, only a tiny fraction of T cells will match. Most naive T cells are irrelevant to that specific antigen. They may be useful for something else someday, but they are not the right clone for this particular threat. So when an antigen enters the body, the immune system has to solve a search problem. It has to find that rare T cell whose TCR can recognize that specific peptide displayed on that specific MHC molecule. That is why diversity matters. The immune system is not carrying a giant army of identical cells waiting for one pathogen. It carries many different small populations, each with different specificity. When the right peptide MHC complex appears, the matching clone can be selected. Where does that search happen? A major site is the lymph node. Dendritic cells and other antigen-presenting cells capture antigen in peripheral tissues. They process that antigen into peptide fragments and load those peptides onto MHC molecules. Then they migrate through lymphatic vessels to the draining lymph node. That matters because lymph nodes are the meeting places. They bring together antigen-presenting cells and a large number of naive lymphocytes. The APC is essentially carrying evidence from the tissue to the place where rare matching T cells are waiting. That help solves the search problem. Instead of naive T cells wandering randomly around every tissue hoping to bump into their antigen, APCs bring antigen to an organized immune hub. Inside the lymph node, many naive T cells scan APCs. Most do not match. But eventually, if the right T cell is present, its TCR recognizes the peptide MHC complex. And that recognition starts clonal expansion. Now let's get to the heart of clonal selection. The T cell receptor checks the peptide MHC complex. If the TCR does not match, the T cell moves on. If the TCR does match, the T cell is selected. Why is the word selection so important? The antigen does not create a brand new T cell from scratch. Instead, the antigen selects a T cell that already exists in the naive repertoire. That selected T cell is the clone that can respond to this antigen. But recognition alone is not the whole story. TCR recognition identifies the correct clone, but the immune system still needs to decide whether a full response is appropriate. That is important because T cells are powerful. Activating the wrong cell or activating a cell under the wrong conditions can cause harm to the body. So the immune system uses additional signals to decide whether the selected T cell should truly activate and expand. That brings us to co-stimulation. Naive T cells usually need more than one signal. Signal 1 is antigen recognition. That happens when the TCR recognizes a specific peptide MHC complex on an antigen-presenting cell. Signal 1 tells the immune system, this is the clone that matches. But signal 1 alone is not enough for a strong naive T-cell response. Signal 2 is co-stimulation. A classic example is B7 on an antigen-presenting cell binding CD28 on the T-cell. You can think of signal 2 as permission to respond. If both signals are present, the T-cell can activate. It begins producing cytokines such as IL-2, increases survival signals, and prepares for proliferation. Now, if signal 1 occurs without appropriate co-stimulation, the T-cell may become anergic or fail to mount an effective response. That's a safety feature. The immune system does not want every weak or inappropriate antigen encounter to trigger a full inflammatory response. So here's the simple pattern. Signal 1 selects the clone, signal 2 gives permission to respond. Both together help trigger effective T-cell activation. Once the correct clone has been selected and activated, it begins to proliferate. This is clonal expansion. The original T-cell divides again and again, producing many daughter cells with the same antigenic specificity. That is a key point. These daughter cells are not random. They're copies of the selected clone so that they can recognize the same peptide MHC target. This expansion is essential because the starting number of antigenic-specific T-cells is very small. One rare matching T-cell is not enough to control an infection. The immune system needs numbers. IL-2 and other signals support proliferation and survival. As the clone expands, many of those cells differentiate into effector T-cells that can help fight the infection. Some cells may also become memory precursors, setting up faster responses in the future. So clonal expansion turns specificity into strength. A rare match becomes a large antigenic specific force. Selection plus activation plus proliferation equals clonal expansion. This brings us to clinical timing. Primary T-cell responses are not immediate. That delay is not a failure of the immune system. It reflects the steps that must happen before a strong adaptive response appears. First, the rare matching naive T-cell has to exist. Second, antigen has to be captured, processed, and transported to the lymph node. Third, an APC has to present peptide MHC to the matching naive T-cell. The T-cell needs activation signals, including co-stimulation and cytokine support. Fourth, the selected clone has to proliferate into enough effector cells to matter clinically. All of this takes time. So when we say primary adaptive response takes days, this is why. The immune system is not starting with a huge army of cells specific for every pathogen. It starts with very rare specific clones that build an army after antigen recognition. That has a major clinical implication. Early in infection, the innate immunity buys time. Adaptive immunity brings specificity and power, but it has to go through selection, activation, and clonal expansion first. The takeaway message is primary response takes days because the immune system has to find the right clone, activate it properly, and expand into enough effector cells to fight. Thanks for watching. I'm Christopher Lee, and what you understand today helps you protect patients tomorrow. And I'll see you in the next video.